Hello everyone and welcome to Instant Biology by Dr. Nila. Today I am here with you with another exciting video on a new topic, and this is an important topic in the area of molecular biology. I would be talking about base excision repair in today's lecture. You would be knowing that I have already started making lectures in molecular biology in English also. So in the previous lectures what we have talked about in the repair system, I have already talked about the direct repair system, and in direct repair system I have given you the example of DNA photolyase, and also I have given you the example of O6 methyl guanosine transferase also. So that is what we have completed up till now. Up till the direct repair system we had completed, and today I would be starting with indirect repair system. So in this particular lecture, as I said, I would be talking in English, and this would be solely dedicated to base excision repair topic. And apart from that, base excision repair, we would be dealing with the reason of or how the base excision repair occurs. We would also be talking about the mechanism in the form of diagrams and in the form of flowcharts. We would be talking about the enzymes that are required in base excision repair, and finally I would be taking some PYQs also, so that your concept can be reinforced. So let us start without much ado about base excision repair. Again, for those of you who understand Hindi better, I've already made a video on base excision repair in Hindi. You can go to my molecular biology playlist and check out that video. So let us start. So this is base excision repair, and first thing first, that we are talking about indirect repair system over here. Let me just tell about that. In case of indirect repair system, what is the difference between indirect repair system and direct repair system? This should be clear in your mind. In direct repair system, you do not need to break the phosphodiester bond. That means the sugar phosphate bond does not need to be broken in case of base excision, in case of direct repair system. But today I would be talking about the base excision repair. In base excision repair, it is a type of indirect repair system, and always in case of indirect repair system you would be observing that the phosphodiester bond or the sugar phosphate bond is broken, and then only the repair mechanism functions, right? So this is the basic difference. Apart from the base excision repair, there are many different types of indirect repair system. We would be talking about the nucleotide excision repair also. We would be talking about the mismatch repair system also, and other types of repair systems that come under indirect repair system. This would be our first indirect repair system mechanism, and we would be dealing with base excision repair, as I've already said. So the first thing that you can see on board is, in case of the in case of the base excision repair, which comes under indirect repair system, you can see that phosphodiester bonds are broken for the repair of the lesions. The phosphodiester bonds are broken. Now, for those of you who do not understand what phosphodiester bonds are, these bonds are the sugar phosphate bond. You might be knowing about the sugar phosphate backbone that is present inside the DNA as well as the RNA. So that the bond that is actually forming in between the nucleotides, that is called as the sugar phosphate bond, and it is also mentioned as COPOC bond. COPOC bond. C is this carbon oxygen phosphate, oxygen carbon. It is also referred to as phosphodiester bond. Phosphodiester bond. Okay, so phosphodiester bond is broken in case of indirect repair system. This is what I wanted to start from. Now let us talk about excision repair. We will be coming down and filtering up to the base excision repair. Firstly, we have started with what is indirect repair system. Now dealing with excision repair. What do you mean by excision repair? Excision of a nucleotide or segment. Why is it called as excision repair? Because we are talking over here about excision of a nucleotide or segment of the polynucleotide containing a damaged site. That means one nucleotide can also be taken care of, or if one nucleotide is damaged, that can also be repaired, and if a stretch of nucleotide is also damaged, that can also be repaired using the excision repair system. The condition is again that there should be a damaged site, right? Next, followed by resynthesis of the correct nucleotide sequence by DNA polymerase. Now, this is again an important point. When you are repairing something, that means first of all you have to remove the damaged thing, and then you have to reintroduce the correct segment. Either it is a nucleotide, you have to remove the nucleotide and then bring in a proper nucleotide. That is what you can do. Apart from that, is if a stretch of a nucleotide is damaged, then you have to remove that stretch and then bring in the correct copy of the nucleotide. So the correct copy is always produced by DNA polymerase enzyme. If you are obviously DNA repair, we are talking about DNA repair system, so obviously the DNA will be damaged, and when you would be repairing the DNA, that would be done by the DNA polymerase. Apart from that, there are several other enzymes that we would be talking about as the lecture progresses. So I hope you would have understood this particular slide. You can take an image of this, and so that we can move forward. Okay, so I believe this is clear to you. Let us move forward and talk about the base excision repair, which is today's topic. I was just funneling down to the base excision repair. I started with indirect repair system, then I talked about excision repair, and then finally I'm talking about the base excision repair, which was actually today's topic. So now base excision repair involves the removal of the damaged nitrogenous base from the nucleotide. Now, if you talk about nucleotide, what do you mean by a nucleotide? Nucleotide has several components. Let me just quickly draw it for you. So suppose this is a sugar. Let us say this is, because we are talking about DNA repair system, so this is deoxyribose sugar. For convenience, I'm just writing it as sugar, but you have to consider it as deoxyribose sugar since we are talking about DNA. So this is sugar. On the sugar, there are two things that are attached. One thing is nitrogenous base. Nitrogenous base is attached, and apart from that, a phosphate group is also attached. So this is called as basically nucleotide. That means nucleotide has a sugar. It has to be deoxyribose sugar. Nitrogenous base. This nitrogenous base can be anything like adenine, thymine, guanine, or cytosine. This will be seen, and there will be a phosphate group. Again, this phosphate group will be involved in the formation of phosphodiester linkage, and this nitrogenous base, the bond that is over here, that means the sugar and the nitrogenous base bond, this bond is actually called as glycosidic bond. So I'm going to write it over here, this bond over here. This bond is called as glycosidic bond. Glycosidic bond. Let me just write it down over here. Glycosidic bond. Glycosidic bond. Okay. Now, so this involves the removal of damaged nitrogenous base from the nucleotide. That means I have to remove this damaged nitrogenous base. Suppose this base is damaged or this base is not correct. What will be done is this base, nitrogenous base, has to be removed from here. So this is going to be done, and cleavage of the sugar phosphate backbone at the A basic site. Now, so the first thing would be removal of this nitrogenous base. The first thing would be this removal of this nitrogenous base. If the nitrogenous base has been removed, you can see if the nitrogenous base gets removed, then what will be left? Only the sugar and the phosphate, right? Sugar and phosphate will be left. Remember, the phosphodiester bond is still present because the sugar and phosphate has not been changed. They are not modified. The only thing that has been removed is nitrogenous base. So that site from where the nitrogenous base has been removed, that site is called as A basic site. A basic site. A means removed. Base means the base from that particular site has been removed. That is why it is called as A basic site. And then finally, after the A basic site has been created, then after that the phosphodiester bond is also cleaved off. So this is the second process. The first one is over here, removal of the damaged nitrogenous base, and the second one is cleavage of the sugar phosphate backbone at the A basic site. So this would be the A basic site, and the sugar phosphate backbone over here, it will also be cleaved. Sugar phosphate backbone will also be cleaved. I'll be showing it to you with the help of a diagram, proper diagram. and you will have a better idea about this in just subsequent slides. So again, the first thing was removal of the nitrogenous base, followed by cleavage of the sugar phosphate backbone at the A basic site, and then finally resynthesizing it with the help of the DNA polymerase enzyme. So this is the complete process in short. Okay. Now we would be talking about the detailed process as well with the help of a diagram. Now, damages that are repaired using the base excision repair. What are the damages that are repaired using this kind of repair mechanism is, first thing is minor damages. That means damages that are not very serious are repaired by base excision repair. So example, alkylation and deamination. You might be knowing that at times alkyl groups such as methyl groups are attached at different bases. That can be removed using base excision repair. Or deamination. Now you might be knowing that deamination of certain nucleotides or nitrogenous bases rather can occur, giving off some different types of nucleotides. That can also be done. That means deamination can also be corrected, and alkylation can also be corrected using this base excision repair module. I hope this is clear to you, and you can take a quick image of this slide so that we can move forward. Okay, so I hope this is clear. Let me move forward with the next slide. Now, the first enzyme that I would want to talk about over here that is used in this case is the DNA glycosylase enzyme. Now remember this, it is of great importance. DNA glycosylase initiates repair process. That means this is the first enzyme that you would observe in base excision repair. Now what do you mean by glycosylase? Of course, you would now remember that glycosylase, what it will do is it will cleave the N-glycosidic bonds. What do you mean by removal of this N-glycosidic bonds? N-glycosidic bonds simply are the bonds through which the sugar was connected to the nitrogenous base. Remember, I just showed you a diagram, a quick diagram in which I showed you the sugar was there, the phosphate group was there, and the nitrogenous base was there. So the bond between the sugar and the nitrogenous base was called as glycosidic bond, and this DNA glycosylase, what it will do is it will be cleaving the N-glycosidic bond, and because of this, the removal of the altered base would be there. As I said, that the first process of this base excision repair would be the removal of the altered base, or the removal of the nitrogenous base that was damaged in some way or the other. So what will happen, the damaged base would be removed using the DNA glycosylase enzyme, and next thing is generation of apurinic or apyrimidinic site. Now why are we calling it apurinic or apyrimidinic site? Because definitely the nitrogenous base would be anything. Nitrogenous base, of course, would be either purine, or it will be either pyrimidine. It can be either purine or pyrimidine. Now what are the purine nitrogenous bases? You would be knowing adenine and guanine are purine, and pyrimidine are cytosine and thymine. I'm not writing uracil over here because I'm talking about DNA. So that is why if the damaged nitrogenous base was purine, then it will be called as apurinic site, since you have removed that base. If it was pyrimidine that was damaged and you are removing it, the site will be now called as apyrimidinic site. This is what will be seen. Now, so generation of apurinic and apyrimidinic site, or it is also called as AP site. Next, multiple DNA glycosylases are available. Remember this, that there is no one DNA glycosylase. There are a lot of different types of DNA glycosylase present, and you can classify them on the basis of their activity or the roles that they play. So DNA glycosylase can be monofunctional. That means, as the name suggests, that they would be only having one function, and they can also be bifunctional. That means they can have two functions. We would be talking about these two different enzymes in the upcoming slides. So you can quickly take an image of this. Okay, so I hope you would be done. Now let me move forward. Okay, so now let us look at this image and get a better understanding about how base excision repair functions. So quickly let us see that this is over here the image of the sugar phosphate backbone that I was previously telling. Let me just give me a minute. So yes, now. So if you check this out, so this is the sugar phosphate backbone that I was telling you. This is the phosphate. Remember, phosphate is always present at the 5 prime end, and then over here this is a nitrogenous base. You can see this is again a sugar, over here sugar. This is again phosphate. This is again nitrogenous base, and this is again sugar, something like this. So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So this is the sugar phosphate backbone, and on the sugar phosphate backbone the nitrogenous bases are attached, as you can see. If you pay attention over here, this nitrogenous base, if you remember the structures of the nitrogenous base, you will observe that this nitrogenous base does not belong to the DNA strand. It is actually uracil. And uracil, why or how has uracil come over here? You would remember that uracils are formed as a result of deamination. So this uracil is actually formed due to the result of deamination. And if you remember that I told you, base excision repair caters or addresses repairs of the deaminated bases. So this uracil has to be cleaved from here because it does not belong over here, and the bond that is present over here is is glycosidic bond. And if you remember what I had talked about in the previous slide, I told you that this bond or the glycosidic bond can be cleaved by an enzyme called as DNA glycosylase enzyme. So what will happen then in the next step? You can see that this bond has been broken, and the name of the enzyme, as mentioned, is glycosylase enzyme. So this enzyme, DNA glycosylase, what it will do is it will remove the uracil, and so you can see that the uracil has been removed from here, and now this site is not having any base, or you can see that the base is missing over here. If the base is missing, then it will be called as AP site. AP site, as I previously mentioned, AP site. And so in this way, you can see that how glycosylase will function, and it will remove the faulty nitrogenous bases. Now, let us move on to the next thing that I showed you, that how glycosylase will function. So glycosylase function has been shown over here, and the removal of this glycosidic bond has been shown over here by glycosylase. Now, there is one more enzyme that has been shown over here, and the name of the enzyme is lyase enzyme. Lyase enzyme is also a type of enzyme that can break the bonds in a particular structure. So over here you can see that the O was connected to this carbon. You can see that over here there was a carbon and to which the O was connected, and this bond has been broken down. You can see that the bond over here has been broken down, and since the bond has been broken down, this again has been converted into NH. So this is the structure, this is the function of lyase. Lyase goes one step ahead and breaks a bond. It can break a type of bond, but again you can see that in case of lyase you do not require water. That means it is different from hydrolysis. Water is not required over here. So now you can take the image of this if you want, so that we can move forward. Okay, I believe you would have done that. Now let us move forward to the next slide. Now I was telling you about the DNA glycosylase enzymes. So there are two different types of DNA glycosylases depending upon the function of the glycosylase enzyme. You can classify it into two parts. The first type of DNA glycosylase was the monofunctional DNA glycosylase, and the second type of DNA glycosylase was bifunctional DNA glycosylase. So monofunctional DNA glycosylase, I've already told you that it will have only one type of activity, and what it would be, you can see that only glycosylase activity is seen. Okay, so when only glycosylase activity is seen, you require another enzyme for one more function. One additional enzyme would be required over here, and the name of the enzyme would be AP endonuclease enzyme. Now, what is this AP endonuclease enzyme? It cleaves the DNA backbone 5' to the AP site. Now, remember when I was telling you about the process or the mechanism of base excision repair, I told you that base excision repair would be completed in two different steps. The first step would be the creation of apurinic or apyrimidinic site, and the second step would be the cleavage of the phosphodiester bond close to the apurinic or apyrimidinic site. Now, since DNA glycosylase has already performed its function, or it has already created a apurinic or apyrimidinic site, then we have to move towards the AP endonuclease, and this will be performing the secondary function, that is the cleavage of the the phosphodiester bond that is close to, or that is 5 prime to the AP side. That means you would be knowing that 5 prime direction means, let us say that this is the sugar phosphate, this is the DNA strand, and over here the sugar is connected to phosphate, sugar, phosphate, sugar, phosphate, something like this. And let us say to this sugar a nitrogenous base was connected, and this was 5 prime, this phosphate, this is 5 prime residue, and this is 3 prime. Now what will happen to this nitrogenous base has been removed, and since nitrogenous base has been removed, this has become a AP site. If AP site is this, then what will happen? 5 prime to the AP site. So this site was AP site, 5 prime to the AP site. Over here somewhere the AP endonuclease would be cleaving, and since AP endonuclease would be cleaving, what will happen? The phosphodiester bond would be broken down. So this is what is going to happen after the monofunctional DNA glycosylase enzyme functions. AP endonuclease will come. Now talking about the bifunctional DNA glycosylase enzyme, now you would understand how or where the discussion is actually going. In case of bifunctional DNA glycosylase enzyme, the DNA glycosylase has two kinds of functions. The first function again is the glycosylase function, and the second function is actually lyase, AP lyase. Now I told you what lyase does is breaks bonds, right? So DNA glycosylase functionality would obviously be there, but it will also be having a AP lyase activity, and it cleaves the DNA backbone 3 prime to the AP site. Remember, if a bifunctional DNA glycosylase is present, the cleavage would be at 3 prime end, and if the monofunctional DNA glycosylase is present, the cleavage would be at the 5 prime terminal of the AP site. This is the basic difference, and you should have it in mind because this can be asked in the CSIR net examination. This is a minor difference, but again, it can create a major impact. So this was about the monofunctional DNA glycosylase and bifunctional DNA glycosylase. You can take an image of this so that we can move forward. Okay, now let us move forward and discuss what will happen after the activity of the DNA glycosylase enzyme, AP endonuclease or AP lyase for the fact of the matter. Now results in a gap. Now after the phosphodiester bond has been broken, what will be achieved over here? So you can see that 5 prime deoxyribose phosphate and a 3 prime OH will be created definitely. Now suppose this was the a DNA strand, and the phosphodiester bond has been broken. So what will happen if I write it like this? So the phosphodiester bond has been broken. Over here there is a cut. You can see that this is a 5 prime end, and this is 3 prime end, this is again 5 prime end, this is again 3 prime end. So this is 3 prime OH that will be created, this will be 5 prime phosphate that will be created. So phosphate and 3 prime OH both are present, you can see that. So it will be creating a 5 prime phosphate and a 3 prime OH. Now this gap can be repaired in two ways. Remember, this is an important point that many students do not understand, that in case of base excision repair also, it's not about only cleaving a particular base and removing it, or only one particular site can be addressed. It's not like that. A long patch can also be repaired with the help of base excision repair. So that is why I'm going to tell you about here about this, that the gap that has been created, it can be repaired in two ways. It can be repaired with the long patch repair system, and it can also be repaired using the short patch repair system. How this is done, let us quickly go through this. So DNA polymerase delta and epsilon plus pCNA ring and RFC are used. Now this is the DNA polymerase ring and loading portion, pCNA ring and RFC basically. Now DNA polymerase delta and epsilon, you might be knowing that these are the main DNA polymerases associated with eukaryotic replication. You would be knowing that if I talk about the leading strand, then it is repaired by DNA polymerase epsilon, and if I'm talking about the lagging strand, it is repaired by DNA polymerase delta, and pCNA and RFC are DNA polymerase patch and patch clamp basically. And now 2 to 8 nucleotides added to the 3 prime end. Now what will happen? I'll be showing it to you with the help of a diagrammatic representation also, but in case of long patch repair, 2 to 8 nucleotides will be added at the 3 prime end. That means a long stretch will be added, and simultaneously displacing 5' deoxyribose phosphate to form a flap. Now, how is this going to be done? So remember, so let us say that this is a 3' OH, this is a 5' end, okay, and this is the portion where the gap is created. So this is where the gap is created, and as I said, this is the 3' OH, okay. So 5' phosphate, 3' OH, 5' phosphate, 3' OH, something like this. So how this will be done? So now the nucleotides will be started to add over here, because they are always added at the 3' OH end. So this will be removed like a flap, and this will be added like this. In this way, it will be added like this again. So simultaneously, the removal like a flap will be there, and addition of the phosphates will be like this. So this is what is happening over here. This is what is written: simultaneously displacing the 5' deoxyphosphate to form a flap, and cleaved by flap endonuclease. And finally, what will happen? This flap that would have been created, it would be removed by cutting by flap endonuclease. So this would be cut down by flap endonuclease, and it would be removed. So this is what will happen over here. So this is how long patch repair functions. I will be showing it to you with the help of a diagram. Also, you can take an image of this. Okay, now you can also do the repair system or repair by short patch repair. Now obviously short patch repair would be for repairing of only a small area on the DNA. So how this can be done? Conserved repair polymerase would be used over here, and DNA polymerase beta is the main enzyme. Like in the previous example, we were talking about a long patch repair. We were talking about DNA polymerase delta and epsilon. But over here, we are using DNA polymerase beta. So this is easy to remember that DNA polymerase beta basically stands, or you can correlate beta with base excision repair. So this question can be asked in your examination, that which DNA polymerase is associated specifically with short patch repair system. So DNA polymerase beta, base excision repair, inserts a single nucleotide. It will be inserting a single nucleotide only, because this is a short repair system. Short repair would be there. Inserting a single nucleotide, contains an intrinsic 5' deoxyribose phosphate lyase activity. Now over here again, so DNA polymerase beta is very special. What speciality does it have is that DNA polymerase beta inserts a single nucleotide. It will be inserting just one nucleotide, contains an intrinsic 5' deoxyribose phosphate lyase activity. So 5' deoxyribose phosphate lyase activity I had shown you in case of the previous diagrammatic representation, where I was showing you how lyase would be functioning. So this is how the lyase will be functioning, or 5' deoxyribose phosphate will be cleaved over here. Lyase, again I told you that it will be involved in the breaking of bond, and 5' deoxyribose phosphate. So if you remember the structure of the sugar that I had shown you, it had 5' phosphate in that, and so that bond, or basically phosphodiester bond, will be cleaved by this lyase enzyme. So it removes the 5' deoxyribose phosphate. That means the phosphate that was attached to the 5' carbon of the sugar, that will be broken down and cleaved. And then finally DNA ligase enzyme would also be involved, because you have to seal the remaining nick in both the pathways. I mean, whether you are going for long patch repair or whether you are going for short patch repair, in both the cases you will have to use the DNA ligase, because after the repair system you have to attach, or you have to join the two strands. That is why DNA ligase system will be used. Now quickly we would be seeing a diagram, and finally we would be understanding how this is working. Now quickly you can take an image of this, and so that we can move forward. Okay, now let us move forward, and this is the diagrammatic representation that I was talking about since long. So this would be actually telling you how this would be functioning. So you can see that this is a double-stranded DNA. In this double-stranded DNA, you can see that this is a damaged base. This red color base has been shown over here, and this is a damaged base. Now first thing, DNA glycosylase is arriving at this point. What is the DNA glycosylase doing? If you check this out, DNA glycosylase has removed this base, this faulty nitrogenous base has been removed over here, and the sugar and the basically phosphate is present over here. You can see that the sugar phosphate is present. Obviously you cannot see the phosphate over here because the sugar phosphate backbone is intact. Sugar phosphate intact. That is why the phosphate group cannot be seen over here. Yes, of course, the sugar can be seen. In the next, AP endonuclease enzyme is used, and you know now AP endonuclease would be cleaving the phosphodiester bond, and that is why now you can see the sugar phosphate over here, and the bond has been broken. Previously, the sugar phosphate backbone was intact, but over here you can see that the bond has been broken. And now you can just divide it into two parts. This is the short patch repair system over here on the right. This is the short patch repair system, and this is the long patch repair system. Now in case of long patch repair system, as I have already told you, a flap endonuclease would be utilized. Polymerase delta and epsilon would be moving like this, and the flap would be created like this, like this. You can see that a flap is created, and finally the flap would be removed. The flap would be removed by flap endonuclease, and then DNA ligase will be functioning to join the two ends. So this was one end, this was the other end, and both the ends are joined together by using the DNA ligase. In case of the short patch repair system also, the same thing is occurring. Only the only difference that you can see over here is polymerase beta is being utilized, and in case of polymerase beta, you can see that the phosphate group is removed, and then you would be observing that you can see this is the phosphate group that is removed from here, and then DNA ligase is used, and then both the DNA strands are joined together. So this is the short repair system. The only thing that you have to remember is the enzymes that are differing polymerase beta. And in case of long patch repair system, there are multiple things that are taking part. So this was a diagrammatic representation. I hope you made good sense out of this. So the diagrams that I have taken over here are from Benjamin Lewin, and you can take a good photograph out of this, and so that we can move forward quickly. Okay, so I believe you would have got a clear understanding about the base excision repair process. Now I would want to take up a few questions, basically two questions. Not many questions have been I've seen, not many questions have come from base excision repair particularly, but I found out this question. Basically this is related to the thymidine dimers basically, and in the previous lecture, in the lecture where I was talking about the direct repair system, I could not take the previous year questions, but so that is why I have taken it over here. So in the previous lecture I had told you that whenever the thymidine dimers are seen, these thymidine dimers basically are created by UV rays, and these thymidine dimers can also be repaired by DNA photolyase enzyme. DNA photolyase enzyme, and this enzyme basically functions under the blue light. It takes energy from the blue light, and it comes under the direct repair system. Direct repair system. Direct repair system. And therefore the phosphodiester bond in this case are not broken. So I hope you got a good understanding about the question. So among the following, which mutagen induces the formation of thymidine dimers? So which mutagen creates thymidine dimers? You can understand that UV rays are the mutagens that create thymidine dimers. Now let us move on to the next question. The next question is again the basic difference between the direct repair and the base excision repair. Now, obviously you know that in case of the direct repair system and base excision repair system, the basic difference is the breakage of phosphodiester bond. In case of direct repair system, you do not break the phosphodiester bond, whereas in case of base excision repair, it is indirect repair system and the phosphodiester bonds are broken. So you can see that the first option is direct repair restores original structure of altered nucleotide without replacement, while in base excision repair, the section of DNA containing the distortion is removed and the correct base is added and resealed. So this seems to be correct, because in case of direct repair system, the original structure is replaced, repaired. Without the replacement, there is no replacement, because replacement would have occurred if you are removing the phosphodiester bond, but something like that has not occurred. In direct repairs, homologous recombination, no homologous recombination takes place. Direct repair restores original structure by non-homologous. Again, this is not correct. In direct repair system, exonuclease, a DNA polymerase and a ligase, no exonuclease is not used. So the correct option is the first one. This is the difference between direct repair system and base excision repair. This was asked in 2015 CSIR net examination. So I hope this was clear to you, and I hope you enjoyed today's lecture. So If you liked the video, please hit the like button. Do not forget to subscribe the channel, and also tell us that which videos you would want to, which topics you would want to see in the subsequent lectures, so that we can make those. Thank you so much. Have a good day.